This paper focuses on the precision control of adaptive conjugated polymer properties in aqueous environments through a biomimetic sequence templating approach. The authors investigate the impact of steric and hydrophobic contributions within peptide segments on the properties of amphiphilic peptide polydiacetylene (PDA) conjugates. They explore the effects of molecular volume and polarity changes brought about by dipeptide substitution domains on various material properties at multiple length scales, including supramolecular assembly behavior, chain conformation-dependent photophysical properties, cell material interfacing, and bulk electrical properties of PDA films. The authors demonstrate that steric effects predominantly influence the electronic structure and resulting trends in photophysical properties, while the interplay between size and hydrophobicity of individual residues becomes more significant for higher-order assemblies affecting bulk properties. They show that sequence-tunable molecular volume and polarity can be used to rationally modulate PDA material properties across length scales. The paper highlights the importance of peptide templating groups, such as peptides, in driving and influencing the photopolymerization and chromogenic transitions of PDAs. The authors explain that noncovalent interactions within the peptide moieties, such as hydrogen bonding and electrostatic interactions, can be programmed by introducing specific amino acids in peptide segments to support self-assemblies that satisfy the geometric requirements of topotactic DA polymerization. This biomimetic approach offers a facile synthetic approach to form PDAs under aqueous conditions and enables the rational influence of PDA structure and properties through peptide sequence engineering. The authors discuss previous studies that have demonstrated the impact of DA proximity to peptide groups and the differentiation between the impact of amino acid steric effect versus charge on the morphology and absorption of PDA assemblies. However, the interplay between molecular polarity and residue size in PDAs and peptide PDA conjugate systems is not well understood. The authors emphasize the need for more studies to shed light on how to control material function based on the molecular design of biologically relevant templating groups for PDAs. Overall, this study provides new molecular-level insights into the rational design of adaptive PDA materials under aqueous or physiologically relevant conditions. The findings contribute to the programmability of biomimetic conjugated polymers with adaptive functionalities and offer potential applications in areas such as biosensing, tissue engineering, and bioelectronics. C. Showing the polymerization of DA, planar structures, and the torsional impact of charge repulsion on the PDA backbone during major chromatic transitions. The significance of this figure is that it introduces the key components of the peptide DA system and the potential for controlling the conformation and properties of PDAs through sequence variation in the peptide template region. The molecular structures and assembly behavior depicted in this figure provide a foundation for understanding the subsequent experimental results and discussions. Figure 2 is not discussed in the text of the paper. It is possible that the algorithm was unable to find any relevant discussion or analysis of Figure 2 in the paper. If Figure 2 is important for understanding the paper, it is recommended to read the original paper to obtain a summary of the description and significance of Figure 2. Figure 3 is not discussed in the text of the paper. The algorithm couldn't find any reference to Figure 3 in the text. However, the figure is shown in the paper and may contain relevant information that is useful for understanding the research. It is recommended to read the original paper to see the description and significance of Figure 3. Figure 4 of the paper shows the absorption spectra of different peptide PDA polymers at pH 2. The figure demonstrates the significant hypsochromic shifts in the absorption spectra of peptide PD as compared to the absorption of conventional PDAs. The absorption spectra indicate the dominance of the red phase in the peptide PDAs, which is attributed to the steric effects and hydrophobic interactions within the peptide segments. The figure also provides a schematic diagram of the hypsochromic shift, illustrating the changes in absorption maxima for different peptide PDA polymers compared to the absorption of a reference peptide PDA. This figure is significant to the paper as it shows the impact of peptide sequence on the photophysical properties of the PDAs, highlighting the role of steric effects and hydrophobicity in determining the chromatic phase and conformation of the polymer backbone. Unfortunately, Figure 5 is not discussed in the text of this paper. The algorithm was unable to find any description or significance related to Figure 5. It is recommended to refer to the original paper for a detailed understanding of Figure 5. Figure 6 is not discussed in the text. The algorithm was not able to find any description or discussion of Figure 6 in the text provided. It is possible that the figure is mentioned in the paper, but not in the section of the text that was provided. To fully understand the significance of Figure 6, it is recommended to read the original paper. Figure 7 of the paper shows the electrical and optical properties of peptide PDA films. The conductivity of the films, which were made from 1 peptide PDA solutions at different pH conditions, was measured using 2 probe conductivity measurements. The results showed that the conductivity of the films varied depending on the peptide sequence. Films made from peptide PDAs at pH 2 showed higher conductivity compared to films made from the same peptide PDAs at pH 10. The conductivity of the films also varied with the size and hydrophobicity of the residues in the peptide template region. Smaller peptide molecular volume and lower molecular polarity resulted in higher conductivity. The optical properties of the films were also analyzed using hyperspectral microscopy. The films exhibited a bathochromic shift in spectra compared to the absorption profiles of the peptide PDAs in solution, indicating further aggregation in the films. The homogeneity of the chromatic phase in the films varied depending on the residue size and hydrophobicity, with films made from peptides with bulky residues showing lower spectral matching and lower surface homogeneity. These findings demonstrate that the electrical and optical properties of peptide PDA films can be modulated by the sequence design of the peptide template region. Figure 8 of the paper was not discussed in the text. Therefore, the algorithm was unable to provide a summary or any significant results or conclusions related to Figure 8. It is recommended to read the original paper for information on Figure 8. This scientific paper explores the use of biomimetic sequence templating to modulate the properties of chromogenic polymers. The researchers investigated the effects of steric and hydrophobic contributions within peptide segments on the assembly behavior, photophysical properties, cell material interfacing, and bulk electrical properties of peptide polydiacetylene, PDA, conjugates. They found that steric effects predominantly influenced the electronic structure and photophysical properties, while the interplay between size and hydrophobicity of individual residues affected higher-order assemblies and bulk properties. The researchers demonstrated that sequence-tunable molecular volume and polarity can be used to modulate PDA material properties across different length scales. This work provides insights into the design principles for biomimetic conjugated polymers with adaptive functionalities. If you are an author or copyright holder for the work presented in this video and you have any issue with the existence or contents of this video, please let me know.